Welcome to Module 7, Lecture 1, where we'll be looking at Chapter 9, entitled DNA and Chromosomes, before moving on to Lecture 2 with Chapter 10, entitled Gene Action from DNA to Protein. Our focus with this module is on understanding the structure of DNA, as well as then looking at how DNA codes for the instructions to make protein. Up through this point in the term, we've talked about genetic material as chromosomes containing genes that are responsible for control of phenotypic traits transmitted through gametes to future offspring. In Chapter 9, we're going to look directly at genetic material. We'll consider some landmark studies in genetics. There are 5 landmark studies that I'd like to talk about in classical genetics. Then we'll consider the 6 major features, or maybe characteristics, we might call them, of DNA, before moving on to look at how DNA fits into the nucleus. We call this DNA configuration. And finally, we'll finish Chapter 9 with a study of DNA replication, looking at the steps involved in the duplication of DNA. All cells must accomplish 2 general tasks. Double-stranded DNA must be duplicated, and we call this DNA replication. And then the information encoded by the DNA must be decoded such that the cell can then synthesize the necessary gene products. We call that gene expression. For a molecule to serve as the genetic material, it must possess 4 major characteristics. We must be able to see it replicate. We must have the ability to store information. We have the ability to express the information. And then we see variation by mutation. We've already seen 2 of these in the last slide as 2 major cell tasks, replication and storage. But let's talk about all 4 of them generally. And in doing so, we're going to go back in time a bit with this lecture, back to the early 1900s, to look at the major contributions to the foundation of genetics and our current day understanding of DNA as our genetic material. So we need to take a scope of the early 1900s. We can't walk in knowing what we know based on it being the 21st century. But rather, we need to move back in time and accept the idea that we don't know what it is that is contributing to our genetic material. Is it a protein? Is it DNA? Is it RNA? Scientists didn't know. So that's the step we will be taking back as we look at these 5 landmark studies. But for now, let's go ahead and look at the 4 general characteristics. First of all, replication. A fundamental property of all living organisms is the ability to replicate genetic material. Once it's replicated or doubled in amount, and that happens during the S phase of interphase, it can be partitioned equally into 2 daughter cells. And don't forget, in the formation of gametes, we also replicate genetic material, though the process is slightly different in gametes, given what we must do to go from a diploid to a haploid state. We have expression of information. Expression of stored genetic information is the foundational process of building a gene product. The initial event of expression is called transcription, or making of RNA. There are a couple of types of RNA. Our focus for the moment is going to be messenger RNA, or mRNA. Once mRNA is made, we need to then translate it into the gene product, taking the chemical information in mRNA to direct the construction of a chain of amino acids, which ultimately folds into a protein. We also have storage. Storage of information requires a molecule to act as a repository of genetic information, and that information may or may not be expressed by the cell in which it resides. While we have a complete copy of our genome in each and every one of our cells at any point in time, our cells are only going to express a part of our genetic information. And then finally, variation by mutation. Genetic material is also the source of variation among organisms through a process called mutation. That is, a change in the chemical composition of DNA, which is then reflected during transcription and translation. This ultimately impacts a protein or gene product. Our modern-day understanding of DNA's role in heredity has been made possible by people like Mendel and Darwin in the late 1800s, as well as by countless other scientists in the first half of the 20th century. In this lecture, in addition to looking at the structure of DNA, I want to discuss 5 major studies, landmark contributions. They're classical studies, if you will, that help build the foundation for our current understanding of genetics today. We're going to consider the work of Griffith, of Avery, MacLeod, and McCarty, of Hershey and Chase, of Watson and Crick, and Beadle and Tatum. Our textbook opens with an experiment by English microbiologist Griffith who took one of the very first steps in identifying DNA as the genetic material as he studied pneumonia following the flu pandemic in the early 1900s by looking at a bacterium called Streptococcus pneumoniae, and specifically identifying two colony types, a 2R bacterial colony that was rough in texture and a 3S bacterial colony, smooth in texture. So two colonies of Streptococcus pneumoniae. One looked rough in texture, another colony looked smooth in texture. He took mice, he injected mice with the type 2R bacteria, and they didn't develop pneumonia. We call this an avirulent bacterium, whereas mice injected with the 3S bacteria, that colony, they went on to develop pneumonia and they died. We call that bacterial type virulent. So avirulent didn't cause death and pneumonia. Virulent caused pneumonia and death. Griffith concluded that the capsule shielding the bacteria in the 3S bacteria prevented the immune system from responding and caused a severe infection, whereas those 2R didn't have a capsule. So we saw a colony with a capsule and without, and those with a capsule led to pneumonia and death in these mice. So Griffith decided to try some other experiments. He heat killed both bacterial colony types. And again, he injected mice with the type 2R and then the type 3S. Neither mouse developed pneumonia, neither type developed pneumonia, indicating that heat had killed that type 3S bacteria that was virulent, that normally would cause pneumonia and death. And the heating of that prevented both mouse types from developing infection. That wasn't enough. Griffith then mixed some bacteria together. He took heat-killed type 3 and live 2R bacteria. So now he has the virulent stuff that he's killed. He's going to mix that in with the type 2 bacteria that was avirulent, that didn't cause pneumonia. And you know what he found? He found in this combination of bacteria, all of the mice who were inoculated with this died. They developed pneumonia and they died. So how is it that heat-killed 3S bacteria on their own couldn't kill bacteria, but when mixed with live 2R type bacteria, heat-killed 3S bacteria could result in pneumonia and death? If you've taken microbiology, you know the answer to this, but if you haven't yet taken microbiology, you might be stumped. Paradoxically, analysis of the mice, their blood, revealed large numbers of living 3S bacteria. And as far as he could determine, these 3S bacteria were identical to the 3S strain from which the heat-killed cells had been made. Griffith concluded that the heat-killed 3S bacteria somehow converted live avirulent 2R cells into virulent 3S cells, calling this phenomenon transformation. Again, if you've taken microbiology from me, you know how this works. He further suggested that the transforming principle might somehow be part of that 3S cell. So this was one particular direction of research. Didn't understand what that transforming principle was. We're going to see some other scientists take this a bit further. So ultimately, we move along to this next step in the scientific journey. Griffith's work led others to explore this phenomenon of transformation. And specifically, in 1944, after more than 10 years of pursuing hypotheses, U.S. scientists Oswald Avery, Colin MacLeod, and McCarty laid out evidence that the material responsible that transformed Griffith's cells, the transforming principle, was nucleic acid. So why nucleic acid? Why did they get this? Well, the researchers began with an isolation process. They took 3S virulent cells in liquid, heat-killed them. They centrifuged them down. Then they treated the resulting filtrate with a protein-digesting enzyme called a protease and an RNA-digesting enzyme called a ribonuclease. So they are trying to figure out, do we have genetic material based on proteins, based on RNA, or potentially based on DNA? Protease and ribonucleases are going to break down proteins in an RNA. These enzymes were meant to destroy the activity of a protein or an RNA. However, when adding the enzyme-treated 3S to a living avirulent 2R bacteria, they still found transformation activity. That is, they were still able to convert live 2R type bacteria to a live 3S bacterium with introduction of this heat-killed 3S. In this manner, Avery and his team were able to rule out protein and RNA as the transforming principle of Griffith's. Next, they performed a similar experiment by which they used a DNA digesting enzyme called deoxyribonuclease to treat the heat-killed 3S virulent cells. Then, when adding the enzyme-treated 3S to the living A-virulent 2R bacteria type, the transforming activity was destroyed. And thus they were confident that the active transforming principle in these experiments was DNA. So again, Avery, MacLeod, and McCarty took Frederick Griffith's experiment one step further by using enzymes to break down either proteins, RNA, or DNA to see what that transforming principle was. What could transform a 2R into a 3S living bacterium? Another major piece of evidence supporting DNA as the genetic material was provided during the study of the bacterium E. coli and one of its infecting viruses, a bacteriophage called T2. We commonly call bacteriophage phage for short, and you'll hear me use the term phage moving forward. Bacteriophage or phage is a virus that infects bacteria, consisting of a very simple protein coat surrounded by a core of DNA. In 1951 and 52, Alfred Hershey and Martha Chase analyzed what happens when the phage infects bacteria. Now, not to ruin the surprise for you, but if you've taken microbiology, you already know what happens with a bacteriophage infection. First, and we see the steps here, first, bacteriophage attaches to the outside of the bacterium. We call that adsorption. Then, a piece of the phage enters the bacterium. This, in the 1950s, was what was unknown and what Hershey and Chase were looking for. What was that piece? Was it a protein? Was it DNA? Or was it RNA? So that piece enters the bacterium. It takes over the cell machinery within the bacterium in order to replicate itself within the host cell. Then, after many replications and the making of many new bacteriophage, we have that maturation process. We go through biosynthesis and then maturation. The bacterium is going to be so full of bacteriophage, the bacterium lyses open, kills the host bacterium while releasing bacteriophage into the environment to go on and infect more bacteria. In the middle of the 20th century, no one knew if the material injected was DNA or protein. So Hershey and Chase sought out the replicating piece to determine if it was protein or DNA. Now, to do so, they used a technique called radioactive isotope labeling. They took radioactive phosphorus, P32, and radioactive sulfur, P30 or S35. And now you need to know, DNA contains phosphorus but not sulfur, so P32 effectively labels DNA. In contrast, proteins contain sulfur and not phosphorus, so S35 will label protein. That's key to this experiment. If E. coli cells are first grown in the presence of either P32 or S35 and then infected with phage, the resulting progeny phage will have either a radioisotope labeled DNA core or a radioisotope labeled protein coat, respectively. These radioactive phages can then be isolated and used to infect unlabeled bacteria. So those are the steps here. We're going to take phage, we're going to grow up bacteria in our P32 and S35. Then we're going to get to this next step. So when labeled phage and unlabeled bacteria are mixed, phage attaches these tail fibers to the bacterial cell wall. This is the process of adsorption before penetration. At this point, the complexes, as we inject something, this mystery something, into these new host cells, the complexes are isolated and subject to this high shear force, kind of like being put into a blender, to strip off the attached phages. We want to get rid of those phages now and look to see what's inside the bacterium. By tracing radioisotopes, Hershey and Chase demonstrated that most of the P32 labeled DNA had transferred into the bacteria cell following adsorption. On the other hand, most of the S35 labeled protein remained outside the bacteria cell. Following separation, the bacterial cells, which now contained viral DNA, eventually lysed as new phages were produced, and the progeny contained P32, but not S35. And so what does that indicate? Because we see P32, DNA contains phosphorus but not sulfur. So P32 effectively labels DNA. So because the DNA contained this radioisotope of sulfur, they were able to interpret the results as indicating that phage DNA enters the host cell, directs phage reproduction. Hershey and Chase demonstrated that the genetic material in phage is DNA, not protein. So that was experiment number 3. We're going to go on and briefly discuss the importance of Watson and Crick, reviewing the classical experiments set forth. We've established DNA as the genetic material of living organisms with the 3 studies we've considered so far. With this in mind, we turn to the actual structure of our nucleic acid, in this case DNA. So in 1953, after reviewing the structures proposed by other scientists of the day, because there were a lot of scientists working on this, and it was kind of a race to space type of a scenario, except the race to determine the structure of DNA. James Watson and Francis Crick proposed that the structure of DNA was in the form of a double helix. There had been other proposals that it was a triple helix, or all sorts of ideas floating around. They proposed the double helix, publishing a short paper in the journal Nature in April of 1953. And I'm going to include that in the module. It's a 1-ish page worth of reading. It's not a lot of reading. I'd like to go ahead and talk about the characteristics Watson and Crick described in 1953. But before doing so, I want to move forward in time just by a few years to consider the work of George Beadle and Edward Tatum. They were from the California Institute of Technology, and they were looking at bread mold, Neurospora crassa, which can be grown on a very simple growth medium. They need very easy, simple nutrients. And from this growth medium, the enzymes of a wild type Neurospora can catalyze all of the metabolic reactions, all the biochemical reactions that are needed to make all of the chemical constituents within that cell. Beadle and Tatum reasoned that mutations might alter enzymes so that they could no longer do their jobs. In that case, they would take mutants of Neurospora that might be found that would potentially be unable to make certain cellular components or cellular compounds, and that could only grow on a medium where those compounds or components were added. We might call that an enriched media. To test their theory, Beadle and Tatum irradiated these cells with X-rays. So we are irradiating bread mold with X-rays, increasing the frequency of mutations of that bread mold, then isolating a number of nutritional mutants. These mutants grew on an enriched medium, but they didn't grow on the minimal media or that just very simple growth medium that we saw our wild type Neurospora growing on. So mutants grew on an enriched medium, but didn't grow on the minimal media, which supported growth of the wild type strain they started with. Beadle and Tatum then performed another test. They wanted to grow mutants out on minimal media with 1 amino acid added. So let's add an amino acid. And so that's what they did here. So we have taken X-rays, we've irradiated these cells, the Neurospora. We now have mutant spores are crossed with a wild type, and we're seeing things grow out on complete media as well as seeing things grow out on minimal media. They go ahead and now perform this next test here. They grow mutants out on minimal media with 1 amino acid added, looking to discover the simplest nutritional supplement that would support a given mutant. Ultimately, this study supported the idea that mutations cause a defect in only one enzyme in a metabolic pathway. In this case, leading to the synthesis of we needed some required nutrient for these enzymes to work properly. What did this do? So their work established the idea that one gene, one enzyme hypothesis, and now it's not quite as simple and clear-cut as that, but their initial proposal was we have this one gene leading to one enzyme, and that was the hypothesis, specifically that a gene's activity is related to the function of a very specific protein. So that was their contribution, and it was pretty big at the time. There have been some modifications to this to really nail things down, because it's not quite as easy as one gene, one enzyme. So we have these 5 different classical studies, landmark studies that help us understand genetics as we see it today. We call those classical studies. All right, so now let's go ahead and move back to the work of Watson and Crick. We're now going to look at the characteristics of DNA. And the first one we're going to look at is the idea that DNA consists of nucleotides. Major feature number 1. So 6 major features. Having established that DNA is genetic material in all of our living organisms and that every characteristic of our cells, from shape to function, is dictated by information held within DNA. We'll go ahead and delve into this first idea. DNA consists of nucleotides. And a nucleotide is made up of 3 subunits: a nitrogenous base, a nucleobase, or just base for short, a pentose sugar, a 5-sided ring, in this case it's deoxyribose sugar, and then a phosphate group, phosphorus surrounded by 4 oxygen. So we see this sugar here, this is our ribose sugar. Ribose just means a 5-sided ring. It is a deoxyribose, meaning we've removed an oxygen from this area right here. We have our nitrogenous base, we have our phosphate group. And I want to point something out real quick. Our pentose sugar here, so here's our deoxyribose. This was the image that we saw on the last slide. If we look at it more closely, we number carbons. There are 6 carbons in this 5-carbon pentose ring. We have an oxygen molecule here, and then we're going to see a carbon at this point. This is carbon 1. We see a carbon here. This is carbon 2 at this point. We see carbon at this spot here. This is carbon number 3. This is carbon number 4. This is going to be, we have the stem sticking out, and then we have this chain, which includes a carbon attached with an OH. So this is carbon number 5. So carbon 1, 2, 3, 4, 5. We're going to see as these nucleotides connect together, how especially carbon number 3 and carbon number 5 play together in joining nucleotides together. All right, now let's go ahead and move on to our second point. Nitrogenous bases are one of 4 types. So we have 2 different major groups of nitrogenous, or of our nitrogenous bases. We have purines and we have pyrimidines, and which have 2 types of their own. So this was what gives us our total of 4 different bases, or nitrogenous bases. We have purines. The 2 purines are adenine and guanine. They are both double ring structures. So we have a 5-carbon ring connected to a 6-carbon ring for adenine. We have a 5-carbon ring and a 6-carbon ring associated with our guanine. We also have pyrimidines. This is our second category of nitrogenous bases. And these, we see only 1 ring structure. We see a 6-sided ring structure, 1, 2, 3, 4, 5, 6 here of our thymine. We see our cytosine also being a 6-sided structure. And they're similar. There are differences here. And we see the differences kind of come about in this area as well as this area here. When we look at our purines, we see some differences in this region as compared to this region. So these are what we see as the 4 different types of nitrogenous bases: adenine, guanine, cytosine, and thymine in association with our DNA structure. Now, moving on, characteristics of DNA, we're looking at the strands themselves. So point number 3, 2 strands coiled around a central axis. We have 2 individual chains or strands of DNA coiled around a central axis, forming a right-handed double helix. And what does that right-handed double helix mean? Well, if you imagine looking down upon this double helix, you will see it spins in a clockwise direction, moving to the right. Imagine a right-handed turn if you were looking down the double helix from above. That's what we mean by this right-handed double helix. Point number 4, here, 2 strands are anti-parallel. The 2 polynucleotide chains are said to be anti-parallel. That is, their orientations run in opposite directions. And what does that mean? Essentially, it would be like putting 2 people together facing in opposite directions. My head facing someone else's feet, their feet facing my head, or vice versa. They're running in opposite directions, and we simply mean that to be anti-parallel. Another way of looking at it is looking at a freeway. For instance, I-5 runs north and south. They're running in opposite directions with numbers as exits or mileposts. So that's what we mean by anti-parallel. And so we're going to see this chain runs in what we call the 3' to 5' direction. This chain, which is going to line up with the nucleobases here, we're going to see that this runs in the 5' to 3' direction. So again, that's what we mean by anti-parallel. Next, point number 5, nitrogenous bases are located inside and they are paired. And what does that mean? Nitrogenous bases of both chains are located on the inside of this double helix. You see here, if we're looking at, well, actually, let's look here first. So we see our bases here. We see our cytosine with our guanine, cytosine, guanine, cytosine. This is hard for me to see in such a small little context. I imagine it is for you too. Guanine, cytosine, cytosine, guanine, thymine, adenine, adenine, thymine. And so we're going to see these found inside the double helix. And this just gives you a better idea here. Guanine, cytosine, adenine, thymine. We call this base pairing complementary. Our purines and pyrimidines are going to pair together with the help of hydrogen bonds. We have 2 hydrogen bonds that form between adenine and thymine. These little slashes represent a hydrogen bond. So 2 hydrogen bonds between adenine and thymine. And then 3 hydrogen bonds here that form between guanine and cytosine. This was discovered even before Watson and Crick's times by a scientist named Erwin Chargaff, an American biochemist who studied a few years before Watson and Crick's discovery, who were studying this a few years before Watson and Crick's discovery. Chargaff noticed that the amount of adenine residues was proportional to the number of, or the amount of thymine residues, while the amount of guanine residues was very proportional to the amount of cytosine residues. So now we know this proportionality to be the result of base pairing. So for every guanine, we need a cytosine. For every adenine, we need a thymine. Okay, so that is point number 5. Point number 6, this is just a bunch of numbers. It's important. It's unlikely I would test you on these numbers, but you should be aware of some of the major concepts. When we look at specific measurements of our DNA strand, we can measure the distances of base pairs apart. So we measure our DNA in a unit called the angstrom, and our bases are roughly 3.4 angstroms apart. So we're going to see that, and we see here's a base here, a base pairing, and here's another base pairing, and these are 3.4 angstroms apart in distance. Further, we see each complete turn of the helix from one point to the next turn, a complete turn. So if this is the start of the turn and this is the finish of the turn, we're going to see that's 34 angstroms. And we have about 10 base pairs per complete turn of the chain. That's absolutely a test question worthy. How many base pairs do we have per complete turn of our DNA structure? In addition to that, the diameter of the double helix, so from the outside point of our sugar phosphate to the other outside point, our diameter is about 20 angstroms. And lastly, our DNA forms what we call major and minor grooves. So here's a major groove, here's a minor groove, here's a major groove, here's a minor groove. Those are going to be 12 angstroms in measurement for our minor grooves, and then we have 22 angstroms for our major groove. Okay, so we have different measurements associated with our double helix, and these were also things that Watson and Crick could work out as they studied this structure, and they put models together before proposing their final list of characteristics in that first publication. As we move from our understanding of the DNA double helix structure, I now want to look at how DNA is configured within the nucleus. By this point, you should have asked yourself, how does all that DNA we have fit within the nucleus of the cell, yet still make itself available and accessible for DNA replication as well as gene expression without becoming tangled or damaged? So let's take a look here. Ultimately, DNA is packaged up very tightly with the help of a protein called the histone. A histone provides structural support for DNA, so we're going to see histone right here. And it provides structural support. Specifically, DNA can wrap itself around histone proteins somewhat like a spool of thread. There's a positive charge to the histone molecule as a result of some basic amino acids, lysine and arginine. making up histone proteins and ensures this strong binding to a negatively charged DNA molecule. The net charge on DNA comes primarily from phosphate groups within that sugar phosphate background. And so we have that negatively charged backbone attracted to the positive charge of our histones. But that alone isn't enough to condense DNA to the extent that's needed. Histones interact together, joining up in groups of 8. So we're going to see that in this next structure here. We're going to see these groups of 8 histones come together to create an octamer that's called a nucleosome, sometimes referred to as the basic unit of eukaryotic chromatin. A string of nucleosomes then coil into even denser structures called the solenoid. And so we have a solenoid. I don't think our textbook mentions solenoids specifically, but that's going to compact DNA even further. And then we have more condensing and compacting of DNA to ensure the genetic material ultimately goes on to fit within the very small volume of the nucleus through the help of loops. So we have looping, and other proteins contribute to this, thus creating chromatin. With all this in mind, we know that DNA in the eukaryotic chromosome consists of one continuous double helical fiber along its entire length, but DNA isn't structurally uniform from end to end. And so this is another concept that I want you to take into account. Rather than being very continuous and compacted, in the early part of the 20th century, it was observed that some of the chromosome remains condensed during interphase, other parts are uncoiled, and with this in mind, the terms euchromatin and heterochromatin were coined to describe the parts of the chromatin that are uncoiled and those that are more tightly condensed. So, 2 basic types of chromatin. Euchromatin is the genetically active type of chromatin that's uncoiled and involved in DNA replication or the transcription process. In contrast to euchromatin, we have heterochromatin, which is highly compressed, very tightly wound, less accessible to the polymerases that are necessary for replication and or for transcription. Now, let's go ahead and trace our genetic material beginning with chromatin. So we're going to move in this direction now. At the bottom, we have this replicated chromosome consisting of 2 sister chromatids. So we have a chromatid here and a chromatid here joined at the centromere. And so that's going to be made up of chromatin fibers that are wound around scaffold proteins. We refer to this area here as looped chromatin. And that's going to give away to chromatin itself before we begin and are able to see those 8 histone unit nucleosomes. We're going to see those here. They have short spans of something that we call linker DNA. So we're going to see our linker DNA here and here, as well as here. From there, we're going to see our individual histones. So these are now histones. We've gone from the nucleosomes that are 8 unit histones now to the individual histone. And then we're going to see our naked DNA here, the double helix itself, all with histones removed and base pairs visible. So the most complicated to the more simplistic view here. All right, let's go into DNA replication. DNA is copied such that as cells divide, the information held within DNA can be passed along to future generations. With this in mind, we go back to Watson and Crick. They recognize that because of the arrangement of our nitrogenous bases, each strand of our double helix could serve as a template for the synthesis of its complement. And so what that means is we can pull these strands apart and we can use each of the original strands to make a new strand by which we have DNA ultimately moving to one nucleus and this moving into another nucleus as we are undergoing cell replication. So Watson and Crick proposed that if the helix were unwound and separated, each nucleotide along the 2 parent strands, so here we have these nucleotides right here, would have an affinity for its complementary nucleotide. In this way, 2 identical double helices would form from one original parent double helix. Each replicated DNA molecule would consist of one old strand and one new strand, and we call this process of DNA replication semi-conservative. DNA replication happens during interphase, specifically, remember, the S phase of interphase. And to understand the steps of replication, I want you to think about the parent strand having to be unwound and separated, hydrogen bonds holding base pairs together need to break. Ultimately, 2 new complementary strands are being built. This is all happening in the nucleus. We need to recruit new nucleotides. Let's look at the details of how this takes place. And what I've done here is I have listed the enzymes and proteins for the process of DNA replication, all serving an important purpose. We'll see how they come into play. And certainly there are more details than this, quite a few more details than this, but this is a general overview of the process. So first of all, we're going to have DNA helicase. DNA helicase, and so our first here, and we're going to see DNA helicase right here in this kind of pinkish lavender structure. DNA helicase arrives first. It's this 6-piece protein structure. It's an enzyme that unwinds the double helix. The 2 strands separate, and each strand is going to serve as a template strand for our semi-conservative replication process. And so we see DNA helicase here. We're going to see a strand here and a strand here. These are our parental strands. This is the semi-conservative part. Half of a new strand is the original strand. We have a replication fork, and so this area is the replication fork, kind of moving kind of in this direction. We can now proceed, that replication process can proceed on those strands because we have separated the strands to make them available for replication enzymes. And the first of which we're going to see single-stranded binding proteins come in. They're going to bind along now these individual strands. They stabilize things. They prevent reannealing or joining back together of our original parental strands. They're necessary because there's a strong draw for the bases of one strand, the bases of the other strand to come together, to be drawn together. It's favorable for those 2 strands to be attracted to one another and reanneal. So to prevent that from happening, we have these very small tetramers called single-stranded binding proteins, which help stabilize the individual strands to keep them from reannealing. We also have primase. So primase, what is a primase? It's an enzyme. We actually need a primer to help attract our DNA polymerase. DNA polymerase 3 is kind of the main event here, but we need to use primase to lay down a little primer, kind of like a foundation. Primase is an enzyme that sets down a short strand of RNA primer segments, and so those are going to be in green. So we're going to have a primase come in, an enzyme come in and lay down a few little nucleotides, some RNA primers, which ultimately allow DNA polymerase to bind so that replication can begin. So ultimately, we need a primase to lay down a primer in order for DNA polymerase to be attracted to and come in and begin the process of DNA replication. Without the primers being set down, we can't actually see DNA replication take place. Now, looking at the main event, DNA polymerase 3, the next step, DNA polymerase arrives. It's attracted to these primers at this replication fork. Ultimately, it interacts with the primer we've just laid down, connecting to these template strands. So we will have a DNA polymerase here. We're also going to have a DNA polymerase here. And we're going to see that this strand is treated as a little different than how this strand is treated. We actually need to pay attention to the direction by which we see strands of 3' to 5' end. Here's our 3' end to our 5' end. And there's going to be relevance to this, and I'll mention in a moment. Replication starts at the 3' end. It always is going to start at the 3' end and move toward the 5' end. We say we read something in a 3' to 5' end, and we would then read this strand 3' to 5', and this is going to be what the problem is. Ultimately, we say that this process of DNA replication, we read up in the 3' to 5' direction, and we are going to write down in the 5' to 3' direction. So we're going to take DNA polymerase, it's going to read these nucleotides, and then it's going to write down, and how it writes, it's creating the complementary strand, which is then laid down in a 5' to 3' direction. Because remember, DNA, or the double helix, is anti-parallel or complementary, and so we need to see a 3' to 5', and then a 5' to 3'. And we're going to see that here too. So our DNA polymerase works directionally. We're creating anti-parallel strands, reading up, writing down, reading up 3' to 5', writing down 5' to 3'. Now we can see that's pretty easy and straightforward here. DNA polymerase just continues sliding along as our DNA helicase continues to open up our replication fork in this direction. That's really easy. We can just keep adding nucleotides from our 5' to 3' direction. However, on this strand here, we're going to move in this direction because here's our 3'. We're moving in this direction. 3' areas here, 5' areas here. That's going to be some trouble because we will move in this direction, but here's where it's opening. So we're opening things up beyond our DNA polymerase that's moving in this direction. So what happens, we're going to run out of a template strand for DNA polymerase to read down. And what needs to happen on this strand, the strand we call a lagging strand, we need to make short pieces of semi-conservative DNA. We're going to need to now bring in a new DNA polymerase and pop it right here. And then as it continues to open up, we're going to need to bring in another DNA polymerase, and we continue moving down. We call that process on this strand here, we call this our leading strand. It's super easy. Things just work out so nice and easy here. But our lagging strand here that moves in the 5' to 3' direction, we need to be reading it 3' to 5'. We're going to have to do this discontinuously as compared to what we see in our leading strand up here. So we have new segments of double-stranded DNA we make on the lagging strand. Here's a segment. We're going to be making another segment until it runs into the primer of a segment that's already been made. These lagging strand segments are called Okazaki fragments. Let me emphasize that. Okazaki fragments. They're found only on the lagging strand, not on our leading strand here. And I want to take a moment and just mention we're going to see another enzyme come in, a ligase come in. So DNA polymerase 3, it's incapable of joining this fragment to this fragment. And so we're going to rely on DNA ligase to ligate these two together. We also have this RNase H or DNA polymerase 1 that's going to come in and take out these little primers as ligase joins these together. So ligase comes in this ATP-dependent manner, joins the sugar phosphate backbones of this small discontinuous or discontinuously produced strands of DNA together, creating a continuous strand. Leading strand, lagging strand, we are creating something via a semi-conservative nature. We're relying on the original strand as part of the template to create our complementary strand. Now, this is going to conclude our first lecture of this module. I'll come back. We're going to talk about the process of gene expression, where we're using our DNA molecule to create a gene product, most often a protein. So that's the next step of this module, and we'll come back with that in lecture number 2. If you have any questions, don't hesitate to reach out. Meanwhile, make it a great day.